seminar. Come on in. Yeah, we're just getting started now. I'd like to begin by thanking Hiroshi for doing a great deal of the work to organize this visit. And I'd like to thank the administrators who were willing to see an opportunity here to bring people together to hear our visitor, Ronan O'Malley. And unfortunately, Hiroshi got COVID, so he'll be watching online. So I'm his stand-in, but I'm very glad to be put in this position to have a chance to introduce Ronan O'Malley. From 2006 to 2016, Ronan was at the Salk Institute with Joe Ecker's group. And many of you know that name, Joe Ecker, a real pioneer in plant genomics, and grabbed a hold of the wave that was made possible by next generation sequencing methodologies. And you associate Joe Ecker's name with that transformation of our business, and you'll associate his name with the tDNA collection that he made high throughput and fully accessible and changed the way our business works. But Ronan O'Malley is the name you should also know, not just Joe Ecker's. Joe Ecker, if you say, thinks a Perl script is something an oyster made, I suppose. The person who is responsible for those big advances is our seminar speaker today, Ronan O'Malley. And after 2016, he went to the Lawrence Livermore labs, the federal DOE lab at the Joint Genome Institute, where he's been since 2016, where he leads the team on sequencing technologies. It's a big group that he's in charge of. And he's known for his mastery and pioneering of sequencing methodologies, but he doesn't just leave it with the long list of A, T, C's, and G's. He also converts those into useful results, and the methodologies end up being able to map transcriptional networks and transcription binding sites and inferring networks from signals you can read out of the sequenced DNA. So it doesn't end with what the sequencer puts out, and that's a distinguishing feature. So where was he before he went to Joe Ecker's lab, and where in 2006? Well, he was here as a postdoc at a very important time in the Department of Botany, as Tony Bleeker's lab was unraveling the ethylene signaling pathway. Ronan was a postdoc here. And then, as you know, the tragedy of 2005, when Tony Bleeker died of cancer, that changed a lot of things and prompted Ronan's movement west. But where was he before he came to us? I remember the day when he visited here. He was at the University of Chicago, where he was working with David Lynn. And if you don't know that name, David Lynn was a really remarkable person, organic chemist, I guess would be the first description, but he plunged into plant biology and did very interesting work on chemical signaling in plants at the University of Chicago, and that's where Ronan also did his undergraduate work. So that's the timeline for the thumbnail of Ronan's arrival here now, and I am just thrilled that you're here now to tell us what you've been up to. And turn on your microphone, and now let's welcome Ronan.
Excellent. Can everyone hear me? Cool. All right, Edgar, thanks so much for the kind introduction. And yeah, let me kind of get full screen here. And so, yeah, and thank you for having me here today to kind of talk about some of my past, current, and also future research directions. So the title of my talk today is Unraveling Gene Networks to Bridge the Genotype to Phenotype Gap. But before I jumped into the body of the talk, I wanted to just give a quick introduction about my lab and the JGIs. It's a little bit different than what maybe people are familiar with normally in academic institutes. I work at the Joint Genome Institute, the JGI, and we're a Department of Energy funded laboratory. So we're part of the larger Lawrence Berkeley National Labs located in Berkeley, California. And I think I would describe this as an advanced genomic science institute. But we're unique in this space where I think the majority or maybe most genomic science institutes are focused on biomedical problems. We're exclusively focused on environmental genomics. And so we deal with topics related to things like climate change and carbon flow, understanding community composition, and then we have a big footprint also in the bioenergy and biomaterials space as well. So as such, the organisms we study are in these plant, fungal, bacterial kingdoms, and we also look extensively at the interactions between them. And so you might guess from the name that genomics is kind of our bread and butter, and this is the case, but we also have very strong metabolomics and synthetic biology groups that help kind of complement our efforts on the functional genomics space. So my team is the Sequencing Technologies Group. It's a team of 35 that includes postdocs, staff scientists, and computational biologists. And really our sort of charge is to develop new approaches to address fundamental problems in genomics and biology. This position really kind of taps into my own background in the intersection of biology, technology, and leadership and team science. So my team is composed of, or my group is composed of 3 teams. Chris Dahm leads our sequencing and high throughput library platforms team. So we have kind of a small fleet of liquid handler robots that we use to produce over 35,000 libraries a year. We heavily utilize our Illumina for NovaSeq for short read sequencing, but we also have a big footprint in the long read space as we have 3 PacBio Sequel 2Es. And the long read is actually almost a full third of our sequencing output every year. So Yuko Yoshinaga is a research scientist in the team. She leads the team that really works on and develops sort of trickier molecular biology-driven experiments, things like ChIP-seq and ATAC-seq. Her group also develops all the libraries for our long-read sequencing. In the last couple of years, we've really been focused on developing single-cell genomics capabilities in the lab. And then finally, Lou Baumgart is a research scientist on my team. He runs what I kind of characterize as sort of the skunks work team. These are these high-risk, high-reward projects that we undertake to develop new genomics methods. And our focus is largely on understanding gene regulatory sequence and the networks that they encode. So broadly speaking, my interest in the focus of my lab is really to try to understand biological variation, really trying to develop systems and approaches that can allow us to start understanding variation, both developmental and how organisms interact with their environment. And so, variation is, just a common theme in biology. We see many just sort of endless examples of variation that can occur. And some examples of variation within species that I'd call out here would be something like a developmental variation in Arabidopsis seed size, where if you look across the 1,000 different accessions that have been collected around the world, there's about a 3-fold difference from the smallest to the largest seed in those wild ecotypes. There's also variation in how organisms interact with their environment. And example of this is how certain sorghum varieties are much better than others at dealing with different stress conditions, in this case, drought stress. And finally, I think probably don't need to explain to anyone how much biological variation can impact our lives, where COVID variants popping up about every 6 months had kept us in kind of this shutdown state for the last couple of years. Now, variation can also occur between species and through either natural or human selection on biological variation within a species. You can end up with really dramatic differences between species, so much so that they may almost be unrecognizable as being closely related. I think a classic and great example of this is the teosinte to maize conversion. So in a course of really only a short period of maybe 3,000 years of selection, this sort of poor nutrient source was now converted into this modern maize that feeds billions of people every day. We also see variation in animals, of course, and in this simple silhouette of, ourselves versus our closest primate cousins, you can see obvious variation in things like body size, body form and gait. And then finally, variation can occur within an individual. So somatic mutations can result in dysregulation of gene functions and result in many of the diseases that we have to deal with, such as cancer. So this is really the phenotype of that, bridging the genotype to phenotype gap that I have in my title. And since the 1950s, we've known that changes to the underlying DNA sequence that is responsible for much of the variation that we see, both between species and within species, as well as within an individual. And these can take the form of single nucleotide variations. They can be small insertions, deletions, or large structural variations. And then collectively, their impact on sort of gene function is ultimately going to be the cause of the phenotypic variation that we see. But maybe for the last 40 or 50 years, it's also been recognized that how the DNA is actually packaged in the nucleus of eukaryotes can impact its impact on phenotype. And so, in the nucleus of a eukaryote, the DNA can be wrapped around these histones and to form nucleosomes, as well as sort of decorated by modifications directly to the DNA basis, most prominently this 5-methylcytosine variant modification. And when the DNA is compacted, you can take a gene, basically silence the function of that gene or expression of that gene. And there are concrete examples where there's no change to the underlying DNA sequence, only a change between individuals and how a specific gene is packaged, whether it's silenced or active, that can result in a large phenotypic difference, such as this agouti mouse that I'm showing on top, or this pyloric mutation in Linear vulgaris. So really to understand where the phenotype and these biological variations we're interested in come from, we need to ultimately be able to understand how this sort of collection of genotypic and epigenotypic changes are ultimately impacting phenotype. So I'd like to start with kind of a concrete example just to sort of get a sense of what does this sort of collective set of changes look like, and what kind of genes are changing and what kind of mutations are occurring. And I think the teosinte to maize is a really great example, as it's been studied for a long period of time and it's well understood in terms of a lot of the players involved in this. So in this this large phenotypic differences between these two species, the very gross phenotypic differences that we see are caused by only differences in 5 genes. And I think a second interesting aspect of this is it appears that all 5 of those genes are transcription factors. And then finally, if we look at the types of changes that we're seeing, the majority of the changes, actually 4 of the 5, are not changing the function of the gene, but they're changing sort of how the gene is being expressed, kind of the when, the where, and how much. And so these are changes not to the coding sequence, but to the non-coding sequence. So I think there's a couple of interesting aspects to this that I want to unpack over the next couple slides. So the first, I think, is this interesting observation. There are a really small number of changes to genes, or a small number of genes can have a large impact, and that, at least in this case of the teosinte to maize, that transcription factors may play sort of this outsized role in biological variation. So transcription factors are this class of protein that bind to specific DNA sequences known as motifs, and then they can act to turn the gene on, or depending on the transcription factor and the binding site, can act to turn the gene off as well. And transcription factors, as its master regulators of these genetic programs, are often impacting the expression of hundreds or even thousands of genes in the genome. So a change to a single transcription factor then can have sort of this cascading effect on the transcriptome. And I think this helps understand why transcription factors may really be playing this particularly important role in the biological variation that we see. I think a second interesting aspect of the teosinte to maize example is that the majority of the changes are actually in the non-coding sequence. And once again, I think this brings us back to the potential importance of understanding transcription factors and understanding biological variation, in that very likely what's happening is that the change to the underlying DNA sequence is impacting the affinity for the transcription factor to bind to its motif. So yet again, it's impacting sort of this cis-regulatory property of the transcription factor. Now, of course, this is just one example, so it's important to look globally. And if we look at meta-analysis of genome-wide association studies of both human and maize traits, we can see that actually a majority, and in fact maybe a vast majority of the changes that are impacting phenotype are actually located in this non-coding region. So, just kind of coming back to sort of this simple model then, if we want to start understanding these causal variants and identifying them, then we're going to need to have, a couple of key pieces of information. We'll need to know where the transcription factors bind in the genome, and then we'll also need to know how a genetic change, or potentially also an epigenetic change, can impact the affinity of that transcription factor to bind to a specific site, and thus impact its ability to control that gene's regulate that downstream gene. But this is a little bit of a complex problem, and I'll talk about why in a couple of slides here. So first off, if we're looking at just a really simple example of differential gene expression between two different Arabidopsis strains, what we can see is that there's over 100 different genetic changes that could be contributing to this. And if we add just the level of methylation to it, there's another couple dozen more potential sites that are contributing to this differential expression. The reality, though, is that it's probably only a very small number, or even just one, that actually is having this impact on this gene expression. So the problem really is this kind of needle in a haystack problem. How do we find this small number, or this small or even one causal variant, kind of this sea of non-causal variants? And then similarly for the methylcytosines, how do we identify that methylcytosine that's impacting the gene regulation as opposed to the ones that are not influencing it? And so once again, though, if we have the information about where the transcription factors bind, and we know how they're either impacted by a genetic or an epigenetic change, it's a solvable problem because we can essentially overlay the two sets of information and identify these potential causal variants. And the problem, this is really at the crux of the problem, is we don't have this information. So for the vast majority of species, we don't know where transcription factors bind, we don't know what their target genes are, and we don't know what the impact of the epigenome variation is on that binding. And so just to give you an example of the 1,800 or so transcription factors in Arabidopsis, only about 20 have very high quality chromatin immunoprecipitation data, or ChIP-seq data. And so this is only about 1% of all the transcription factors. As a consequence, these non-coding regions where we need to know where the transcription factor binding sites are essentially these deserts of information. And so really moving forward, what we need to be able to really start understanding gene regulation and then more broadly these gene regulatory networks is we need to know for kind of a comprehensive set of transcription factors within a species, where do they bind and what are their motifs? If we want to understand the impact of the epigenome, then having information about how DNA methylation impacts that binding is going to be valuable. And then I think finally, really it's going to be critical to understand everything for how an individual gene is regulated, then to how these larger regulatory networks work, is to have these sort of databases of TF target gene relationships for a large portion of the gene TFs within a species. Now, the reason that we're sort of short on this information is that though ChIP-seq is a really powerful technique for characterizing transcription factor binding sites in vivo, it has its challenges. Primarily, it's very labor intensive and it's highly prone to failure. And this is in large part due to the fact that it relies on antibodies that are expensive to generate and often have too low of affinity for the TF ChIP-seq. And then as a consequence of this, it's really a method that's never been effectively scaled, and the only sort of species where we do have these sort of comprehensive TF binding site maps are for really well-funded consortium efforts in human and mouse, such as the ENCODE project. And so really seeing this applied, even to kind of a model system like Arabidopsis is highly unlikely that we'd have this sort of commitment of large amounts of both money and researchers' time to generate these large collections. And then if you think beyond that to the many, many other species that people study, generating these kinds of TF target gene and TF binding site databases is going to be very unlikely to happen, I think, in the foreseeable future. Additionally, even in those cases where we do have this information about the TF target sites in such as human and mouse, the ChIP-seq doesn't have the ability to measure the impact of DNA methylation on transcription factor binding. So even in those cases, we're sort of lacking that information as well. So it's kind of with all of these considerations in mind that I came up with an alternative approach for characterizing TF binding sites that could be applied very broadly and at scale. And so the approach that I came up with is this DNA affinity purification sequencing, or DAP-seq. And so this is an in vitro transcription factor binding site discovery approach. And I'll describe how it works in the next couple slides. So DAP-seq starts by a scientist extracting genomic DNA from a tissue. Then they fragment that and they attach on adapters, essentially creating an Illumina sequencing library. Then in a second step, you take a plasmid that contains an affinity tag and a transcription factor sequence, and then using an in vitro transcription translation kit, you then produce a transcription factor affinity tag protein. You can then immobilize that transcription factor affinity tag protein on a bead that contains a ligand that complements that specific affinity tag. Finally, then you combine the genomic DNA library with this immobilized transcription factor, and then you allow the transcription factor to then bind to its target sequence. The rest of the unbound fraction can be washed away. You elute the bound fraction, you amplify, you sequence it, and then you map it back to the genome. When you see this sort of pileup of reads at a specific location in the genome, or what we call a DAP-seq peak, then you know that's the location where that transcription factor binds in the genome. And as you can see, the DAP-seq peaks look very similar to the ChIP-seq peaks, and so much so that you can use all the same software that's been developed over the years to analyze ChIP-seq on the DAP-seq data set. So using one of those packages, we were able to then call DAP-seq motifs. And what we find is they agree very closely to the known ChIP-seq motifs that have been previously characterized, or motifs characterized through other TF binding site discovery approaches. So I think importantly, though, the DAP-seq is also able to capture much of the information that's present in the ChIP-seq data sets as well. So genome-wide, a comparison of 14 different transcription factors that we had both DAP and ChIP-seq data for, we were able to capture between 40 to 98% of the ChIP binding sites with the DAP data, with an average of about 75% of the ChIP-seq sites present in the DAP-seq data set. So as I said, one of the goals of the method was really to make it both scalable and also low cost. And the DAP-seq really kind of checks both those boxes. So because we're doing an in vitro transcription translation approach, every well can be a different transcription factor, and it's possible to run multiple 96-well plates in a day. It's also a relatively inexpensive approach, so each transcription factor costs about $50 for the DAP-seq experiment all the way through the sequencing as well. So with this really highly scalable and low-cost approach, then we applied this to all 1,800 transcription factors in Arabidopsis, and we were able to get successful DAP-seq data sets for 529, or 30% of those. I think importantly, we were able to get representative we were able to get good, successful DAP-seq for all 70 transcription factor families, as well as representatives from all the subclades within those families. So from this, we believe we have a fairly comprehensive map now of all the potential transcription factor binding sites genome-wide in Arabidopsis. So potentially, maybe not all, but many of the sites that were previously not known. So in this 2016 Cell paper, we published the method. We also provided the full resource of the binding sites, the motifs. And then I think importantly, we also provided this large TF target gene database for this full set of transcription factors. We're able to show that the target genes identified by the TFs could be used to then predict correctly the function of known transcription factors, as well as identify functions for a large number of transcription factors that hadn't been previously characterized before. And so I think this data set, has been sort of adopted fairly generally by the Arabidopsis community, as well as more broadly by the plant community. People have applied it to a lot of different questions related to everything from how an individual gene is regulated to understanding the role of a specific transcription factor they're interested in, as well as other people also using it for sort of this bigger picture, trying to build these gene regulatory networks associated with specific traits. So it's been great to see that the data set's been useful and adopted in the plant community, but I was also really always interested in applying this method sort of broadly. It's not built just for plants, it really could be applied to any species. So I was excited to get an opportunity to collaborate with Louise Glass at UC Berkeley. And so Louise's lab, among other things, is very interested in how the fungus Neurospora makes decisions about which specific nutrient sources to use. And over the years, they'd collected different transcription factors that they could demonstrate through different methods had a role in some of these nutrient choices, but they didn't know what their direct targets were. So in our collaboration, we tested this set of transcription factors that they had, and this has been the basis of these 4 collaborative papers that we published over the last few years. So I wanted to dive a little bit into one of the stories from this recent 2021 PNAS, as I think it kind of illustrates how valuable the DAPSeq data can be in understanding gene regulatory networks. So the fungus Neurospora, when it has its preferred nitrogen sources, nitrate or ammonia, it'll use those exclusively. But when they're not present, it'll then switch its program to use a non-preferred nitrogen source if that's available in its environment. And it was known that the kind of regulation of the processing of these non-preferred nitrogen sources was performed, one, by this general regulator, NIT2, which is present whenever there's a non-preferred nitrogen source being used. And secondly, this would then be paired with a transcription factor that was specific for that nitrogen source that was available to the organism. So based on RNA-seq co-expression analysis, a model had been developed that the general regulator NIT2 was controlling a large set of genes, and then the nitrogen source-specific transcription factor was then co-regulating a subset of those genes to really kind of tailor the response specifically to that chemical. However, when we did the DAPSeq and now had the direct target genes, the story became much simpler. And what we observed was that the direct targets of the general regulator NIT2 were almost exclusively transporters involved in uptake of the nitrogen, while the nitrogen source-specific transcription factors are primarily controlling catabolic enzymes involved in processing it. So we moved away from this sort of co-regulatory network that came from the RNA-seq co-expression analysis, that once we had this direct target information, we can now see that it was sort of this mutually exclusive or sort of distribution of labor that's actually happening here. So I think this is an example of how having this direct target information can really simplify what might seem very complicated if we're only looking at something like RNA expression alone. So in the introduction, I also talked about how I was interested in being able to look at the impact of DNA methylation on transcription factor binding and its potential role in modifying gene expression. So the DAPSeq really is kind of perfectly designed for this. Because we're using genomic DNA, it contains the native methylation pattern of the tissue that we extracted the DNA from. So from this, you're able to then directly determine whether a transcription factor is inhibited by the presence of methylation, preferentially binds to a methylated site, or doesn't care either way. So to help with this, I developed another version of the assay that we call amplified DAP, or AMP-DAP. So in AMP-DAP, what we do is we just add an additional step where we take the input DAPSeq library and we amplify it for 10 cycles of PCR. And so what this does is it creates essentially a methylation-free copy of the input library. We can then run the DAPSeq and the AMP-DAPSeq through the normal process and compare the data sets. And so on the left here, I'm showing a high-scoring motif that doesn't contain any methylation. We get very equivalent binding for both the DAP and the AMP-DAP experiments. On the right, I'm showing a site that contains methylation in the motif. And what you can see is that only the AMP-DAP is showing binding at this site, indicating that for this specific transcription factor, the presence of DNA methylation alone is able to prevent binding at this specific site. And I would note that the ChIP-seq actually tracks quite well with the DAP-seq data set for this transcription factor or for other transcription factors that are inhibited by the presence of DNA methylation. And this would indicate that, though there's other potential processes going on in the nucleus to kind of prevent transcription factor binding, that DNA methylation alone may be sufficient for exclusion of binding for this transcription factor. So then taking a ratio of the DAP-seq and the AMP-DAP data, we were able, kind of genome-wide, then able to identify or able to observe that 75% of Arabidopsis transcription factors are actually sensitive to DNA methylation, with the majority, about 70% of those, inhibited by the presence of methylation, but then a small portion of them actually preferentially binding to a methylated motif. So in this original approach, we could only really sort of generally say whether methylation in a site was impacting binding. But with the newer version of the assay, where we actually bisulfite treat the DAP product after elution, we can now determine directly what the contribution of an individual methylation site is to the affinity of the transcription factor binding. So just kind of in conclusion for that section, I think the DAP-seq has allowed us to really massively expand the TF motif binding site and target genes, both for Arabidopsis, but from the Neurospora example, I hope I've shown that it really can be applied to any species. Additionally, it has this unique ability to be able to measure the impact of DNA methylation on binding. And we were able to demonstrate that over 3/4 of Arabidopsis transcription factors are actually sensitive to DNA methylation. And while most are inhibited, actually a small portion of them preferentially bind to these methylated sites. And then I think finally, more generally, being able to provide these large comprehensive TF target gene databases has been very valuable, I think, for the plant community. And we're looking to sort of expand this going forward so that we have this information really available more broadly across plants as well as other species. So this includes sort of ongoing work at the JGI to apply DAP-seq to create sort of global atlases of transcription factor binding sites for the 7 JGI flagship plants, as well as 5 fungal species. I'll talk a little bit about a project that we've done to apply DAP-seq in bacteria. And finally, we have quite a large number of smaller scale collaborations focused on kind of specific questions in the plant, fungal, and bacterial space. So I think the DAP-seq is a powerful method. I think it's scalable and has a really high quality of information that we can get. But we were always looking to see if there was ways that we could continue to improve on it. And in this recent Nature Methods paper, we published 2 new versions of the assay that allow us to do a couple of things that I think are interesting. So the motivation for this first approach, biotin DAP-seq, was that though running the DAP-seq experiment is a really low cost and fast proposition, actually having these sort of large TF clone collections is slow and expensive to generate. And so we were looking for a way that we could speed this process up and also drop the cost of this sort of upstream step of having these TF affinity tag clone collections. And the second method, this multi-DAP or multiplex DAP-seq, the goal really was that we know that TF target gene relationships are conserved across species, and having this information can be really valuable in terms of understanding sort of core conserved components of a regulatory pathway, as well as understanding sort of how different species may be actually changing their regulatory pathways to deal with sort of specific local conditions or specific developmental programs. And so we were interested if there was a way that we could capture this sort of TF target gene relationships kind of in a scaled and fast way. And so first I'll talk about this biotin DAP-seq method. So as I said, In the DAP-seq, it's really low cost, but this first step of having these large TF affinity tag clone collections, sort of prerequisite that we needed, is very expensive. In total, it's about 85% of the cost, and probably takes substantially longer to actually have the clone collection than it does to actually run the assay. So we were looking to see if there was a way that we could simplify this by just using PCR to generate our input TF template. And so I'll describe then how we ended up solving this problem. So if we start with either genomic DNA or cDNA, we first build primers that target that coding sequence. And then we include a small sequence at the beginning of it that's needed for the in vitro translation step, transcription step, but we don't include any affinity tag sequence in here. We now have many copies of this TF template, and then we add that into our in vitro protein expression mix. We then let that incubate. The DNA is then transcribed into RNA. It's then translated into a protein. And then to get the affinity tag in, what we do is we add in a biotinylated lysine tRNA. So this is a normal lysine tRNA. It just has a biotin moiety attached to the lysine. Then during the translation step, you incorporate at a random lysine positions, about 20% of lysines, this biotinylated lysine, and then this creates this biotinylated tag TF protein. We can then go through our normal DAP-seq process of creating the library, exposing it to the affinity tagged transcription factor, pulling it down with a streptavidin bead, and then after mapping back, the data that we get is really identical to the data that we get from the affinity tag transcription factor plasmid that we would have used. But the advantage here is it's really highly efficient with about a 10-fold reduction in both cost and time. So the second method that we introduced in this recent paper is this multi-DAP approach. So as I said, TF target gene relationships can be conserved for really long evolutionary times. And also, understanding that information can be really valuable in sort of understanding what are the core genes involved in these regulatory programs. Also, I think over looking at sort of how they drift can provide information about how regulatory pathways are, provide phenotypic innovation. And so the approach that we had then to really capture this information in a scaled way is this multi-DAP approach. So in the multi-DAP, what we do is we will take a genomic DNA, we'll extract genomic DNA from multiple species. We'll then label each one with a specific unique molecular barcode, and then we'll pool that together, and that pool then will be the input for the DAP-seq experiment. So just to give you a sense of what we did in this recent Nature Methods paper, in the original Cell paper introducing the DAP-seq method, we had one Arabidopsis genome against 500 transcription factors to produce 500 DAP-seq maps. In this recent study, we had 48 different bacterial genomes pooled together as input against 400 transcription factors, about 200 from E. coli and 200 from Pseudomonas simiae, and this produced about over 20,000 DAP-seq maps. So we selected the 48 different bacterial species kind of in two ways. One, we had species that were closely related to both E. coli as well as the Pseudomonas. So we had this sort of short evolutionary time to be able to kind of compare what's happening, what's conserved, also what's changing in this short period of time. We also then collected different bacterial species that kind of span the entire bacterial kingdom, going back as far as kind of an evolutionary divergence of about 2 billion years. And then this is what the typical results look like. So this is Pseudomonas simiae from this particular multi-DAP experiment was the source of the transcription factor. What you can see is we see kind of very strong binding sites associated with 3 different target genes with a very specific kind of genetic, a binding site architecture. And then through the full Pseudomonas clade, we see that these 3 target genes are conserved, and as well as this specific architecture. And then interestingly, in this very much more distantly related Acidovorax, we can also see that 2 of the 3 genes, and at least part of that binding site architecture is also conserved. So then looking at this then kind of more globally across the larger set of Pseudomonas transcription factors, what we can see is that if we, this green dot here indicates the Pseudomonas simiae, which is the source of the transcription factors. If we see a red box, then that means that the target genes that were identified in this, either close species or more distantly related species has essentially the same TF target genes that were identified in the Pseudomonas simiae, so that we're seeing sort of conservation of TF target gene relationships. And then looking at this sort of for kind of the general patterns, we can see that there's a small number of transcription factors that target gene relationships that have been conserved for very long evolutionary times. We then also see that within the Pseudomonas clade, that the majority of transcription factor target gene relationships are generally conserved across the Pseudomonas genus. And then finally, there's a small number of transcription factors that seem to be more species specific. So the TF target genes are present only in Pseudomonas simiae or maybe in one or two of its closest relatives. So on the conservation side of things, a record holder for the most ancient one was this MRAZ. So MRAZ is a transcription factor involved in cell wall synthesis, cell division. So we see a nearly identical binding motif for both E. coli and for B. subtilis. And they're targeting a very similar set of target genes, but they diverged about 2 billion years ago. So this would put them about half the lifetime of the Earth and would indicate that this TF target gene relationship is present very early in the genesis of bacteria. In addition to conservation, though, we identified cases of plasticity. So one case of plasticity that we observed we called regulatory rewiring. This is when the same transcription factor is controlling different genes but to still execute the same function. So a good example of this is this RSR in E. coli and its homologue in Pseudomonas simiae. So in both cases, the RSR and its Pseudomonas homologue in P. simiae, it's known that they bind to arsenic and that they activate then a response involved in arsenic detoxification. So from the multi-DAP data, we were able to then see that the RSR in E. coli was binding to a set of genes that were known to be involved in arsenic detoxification, specifically a pathway involved in primarily reducing the arsenic to help to detoxify it. We were surprised to observe, however, that the Pseudomonas simiae was targeting a completely different set of genes. So even though we knew that it was still involved in the same function, it seemed to be executing it through a different mechanism. When we looked at the genes themselves, we could see that several of them were metal transporters, and at least one of them had been previously characterized as playing a role in arsenic detoxification of very distantly related species. Additionally, from the multi-DAP, then we were able to see that this pathway 1 is largely conserved throughout the enterobacteria, the E. coli relatives, and this pathway 2 is conserved in this throughout the entire Pseudomonas genus. So a second kind of plasticity that we observed was this regulatory repurposing. This is when a transcription factor and its homologue are binding to, once again, very similar motifs, but controlling different genes, and in this case, to execute different functions. And so an example of this is this ASCG and its homologue in Pseudomonas simiae, where in E. coli, it's controlling genes involved in sugar catabolism, and then in the Pseudomonas simiae, is playing a role in controlling genes involved in defense chemical synthesis. So just to kind of wrap up that section, from the multi-DAP data, we were able to observe sort of patterns of conservation where transcription factors are controlling both the same genes to execute the same function, and this is conserved across evolutionary time. We're also, though, seeing these plasticity cases, such as rewiring with the RSR, where the target gene is controlling, our target genes are different, but they're still executing the same function, as well as repurposing, where we have a different set of target genes, in this case, to execute a different function. And in total, we found about 24 cases of this plasticity of either rewiring or repurposing, indicating that this is a potentially important source of phenotypic innovation for both gene regulation and response in bacteria. So just kind of in conclusion for that section, I think the multi-DAP is a powerful tool for identifying conservation and variation in genetic pathways and networks. And that the biotin DAP really is this useful tool for rapidly surveying transcription factors across many species at low cost. And then since they're used at different steps in the process, they can be actually combined in a single assay that allows you this very ultra high throughput approach for characterizing TF target gene relationships, both conservation as well as plasticity and regulatory networks. So kind of in the interest of time, I'm going to sort of introduce some of my future directions and keep things a little bit quick to just sort of introduce where we are and where we're going right now with things. So, we're currently now applying the plant multi-DAP approach, the multi-DAP approach to plants, and we're using sort of a similar approach where we sort of are targeting a set of TFs in the Brassicaceae, primarily focused on Arabidopsis and some of its close relatives, as well as TFs within sorghum and its close relatives. But we're also doing sort of the reciprocal across the full plant kingdom so that we can start to understand these regulatory pathways and their conservation more broadly. In addition, we're also throwing in some more ancestral species like fern and moss to sort of maybe help us understand sort of where some of these pathways originally emerged from. Now, I think the DAP-seq obviously is an in vitro method, so we're sort of obviously missing this in vivo component. What are the actual available regulatory sites, in a specific tissue, in a specific cell type, in a specific conditional treatment? So we're relying on single-cell open chromatin assays, specifically single-cell attack-seq, to then identify, where are these accessible chromatin regions in a specific set of experiments, or, for example, a time course of drought treatment or hormone treatment, or really sort of any kind of treatment that you could think of. And then I think combining the DAP and the single-cell attack-seq information, I think, will be highly complementary. Additionally, I think complementing this as well, we've been working on single-cell RNA-seq now for a couple of years. We were one of the first labs to publish a single-cell RNA-seq paper in 2019 on Arabidopsis root cell types, and we've been working to really kind of expand this application across a broad set of different species and tissues. And then finally, we're also actively working on single-cell or spatial transcriptomics to ultimately understand where the genes are being expressed, in relation to specific anatomical features. And so really, I think these all fit together quite well, where the multi-DAP can provide us this sort of binding site information. The single-cell open chromatin information can then provide us this sort of ability to infer where are the open chromatin sites, and from the DAP data then annotate those. The single-cell RNA-seq can provide us both information about which specific transcription factors are expressed in a specific cell type at a specific developmental stage at specific conditional treatment to help then further refine our understanding of what's going on in these networks at a cell type resolution. And then I think the spatial then also allows us then to place this into an anatomical feature. Finally, I think the DAP has sort of the methylation rolled into it. So this information could be really valuable for then understanding how differences in methylation between species or between tissues is contributing to this regulatory landscape. So the real goal here is ultimately to build these gene regulatory networks that are cell type specific, anatomically resolved, and methylation aware. And then we're also working on methods for large-scale testing of these networks through protoplast transfection to either understand, disrupt the gene networks at large scale at a large number of genes, or understanding the regulatory potential of promoters also at a large scale, so that we can begin to really not only sort of make the predictions, but then also test and refine these models. So it's really this collection of tools that I see as a potential bridge between this genotype to phenotype gap. I think initially, we'll learn to understand sort of how the genotype and epigenotype is contributing to these different traits and understand sort of their regulatory architecture. And I think ultimately then, with this information, we'd be able to then take directly from genotype or epigenotype information for predicting phenotype, looking forward at kind of precision agriculture or personalized medicine approaches. And so, two sort of biologies that I'm interested in, I'd introduce very quickly. One is seed size, and the Arabidopsis seed size. And this comes from work where I led, as Edgar mentioned, a large-scale project in Joe Ecker's lab to both expand and improve the annotation of the tDNA collection, And also then through a large-scale genotyping project, we isolated homozygous mutants for about 88% of genes. Now, these have been used sort of generally by the Arabidopsis community for all sorts of gene functional testing, but I was always interested in applying this for a really kind of large-scale forward genetic screen, unbiased screen for a phenotype. And so using a flatbed scanner approach, actually through conversations with Nathan Miller, we ended up testing 35,000 homozygous lines for seed size, identifying about 100 genes that are contributing to seed size variation. And so this is kind of a manuscript now that's in preparation with Joe, as well as Liang Song, who's up at, who's a former colleague of mine at the Salk, who's now in Vancouver. And then finally, secondly, sorghum drought response. I became interested in this as I was part of a collaboration between UC Berkely and JGI, looking at this multi-year drought field experiment, really kind of measuring all these different layers of omics, such as RNA-seq, ChIP-seq, and bisulfite-seq. And now I'd like to sort of approach some of these with some of these single-cell multi-omics approaches, as well as with the DAP-seq data to start to understand, specific, traits underlying and the regulatory networks underlying specific drought response traits in different varieties found throughout the throughout sorghum. Kind of generally what this looks like really is, this top path using kind of these cell type resolution assays like single cell RNA-seq and spatial to establish these gene co-expression networks, and then the DAP-seq, the single cell attack-seq, as well as these massively parallel reporter assays to start understanding the gene regulatory networks that control them. Some of the directions I think I'll talk a little bit about in the Chalk Talk tomorrow is, I think the multi-DAP can be a really powerful way for, taking different, looking at variation sort of within a species. And so this can be variation sort of traits such as sorghum drought, within, different plant species such as sorghum. Or I think it is also going to be potential to apply this approach in things like understanding human populations and how genetic variation is impacting the binding site architecture, the binding site landscape, and ultimately potentially impacting the regulatory landscape as well. Addition to that, since we'll be able to use different tissues, we can then also understand what the DNA methylation variation is that is contributing also to this regulatory landscape. I think also, as I mentioned, we're applying the DAP-seq very broadly across the plant tree of life. I could easily see, a project that we would do a similar thing, let's say, across primates or more generally across animals. Once again, both looking at what the genetic impact is as well as what the variation of methylation is impacting it. And then finally, from the methylation side, as different tissues and different cell types are going to have different methylation patterns, testing these in the multi-DAP experiment, either in different plant tissues and organs or in different human tissues, can start to give us some understanding of how the regulatory landscape is shaped by methylation differences. I think in addition to that, applying the DAP-seq even to disease tissues such as cancers, where it's known that DNA methylation can often be disrupted, is a potential also interesting direction to go. So I think kind of this future direction one really focuses on taking sort of traits of interest, either developmental or stress response traits, using the single cell RNA-seq to then characterize what are the individual transcriptome at a cell type level, but also at different developmental stages, combining this then with the spatial transcriptomics to then help both annotate the different clusters in what the different clusters we're identifying with, as well as anchoring those back into an anatomical context. And then the ultimate goal of this is to have these kind of developmentally and cell type specific resolved gene co-expression networks. The second direction really is then looking at the regulatory sequence. So I think combining the multi-DAP to capture sort of these pan-binding site maps, combining this then with the filter of constraints of which sites are sort of accessible and available in different tissues as well as at different cell types at different developmental stages, and then also with different conditional treatments can then provide us sort of the tools that we'll need or the information we'll need to build out these cell type specific TF regulatory networks. And then finally, as these networks will contain thousands of genes and many thousands of connections, it's going to be important to validate these and refine these. And so I've been developing methods with my team for transfecting protoplasts to either disrupt gene function through an approach like Perturb-seq or test many different promoter sequences as well as many mutated versions of them to start to understand where the critical regulatory sequence is involved. I think combining that information with both the single cell attack and the DAP-seq information can then really help us sort of really improve these regulatory networks that we're looking to annotate. And then I think the long-term goal of this is really that once we have a good enough understanding of what are the critical sets of genes as well as what are the critical regulatory networks involved, can we start to then take sets of genes along with their regulatory information and then take traits from one species or one strain and transfer it to another. And so this, I think, really is kind of the long game of all of this work. And so just say in acknowledgement, thanks to my entire team, and I'd specifically call out Leo and Yu, who really led the multi-DAP work, Maxim, Peng, and Yuko, who have been driving the single cell genomics approaches, Matt Lowe and Sharon Greenblum, who work closely with us on analysis, as well as Louise, who I collaborated with on the Neurospora work. And with that, I'll take any questions anyone has.
I'll let Ronan handle pick his own questioners. There's one right away.
So there's more than enough for you to chew on. So far be it for me to suggest something else. But I wonder if the in vitro translation of the transcription factors leaves out useful information if you would purify your tagged transcription factors before a DAP-seq or a multi-DAP or something. Especially as you get into like a multi-DAP where you're focusing in presumably on a smaller number of transcription factors.
Yeah.
They might then have phosphorylations or glycosylations or whatever.
Mm-hmm. Yeah, I mean, it's an interesting possibility that you could, you'd have to get down to a smaller number because then at that point, then you're dealing with an in vivo system. You kind of lose that scalability. But I think, if you were able to then, just even so one version of this that we've been exploring and we're kind of early days on it is to actually float the transcription factor into an isolated nucleus or take the transcription factors and transfect them with the tags so that we can kind of essentially do like an ex vivo chip-seq or kind of an in vivo DAP-seq, I don't know, some version between the two of them. So that's an approach that we're exploring right now. We're kind of early days on it, but it's something that, I think it potentially would allow us to capture kind of other co-binding events, if there's other modifications such as phosphorylation, and then it should also, it potentially also get at the question kind of about the chromatin interaction as well. We're kind of also coupling that then with a nucleosome, an in vitro DAP-seq with a sort of a nucleosome that we've, basically DAP-seq with nucleosome and seeing how that impacts binding. So that is something that we're working on right now, but it's early enough I didn't really want to talk about it. So, so we'll see if it works. Yeah.
So this is kind of related maybe in terms of the transcription factors that need to be in a complex.
Mm-hmm. Yeah, the assay works fine if you throw in the two or multi. We've only done it with two transcription factors at the same time. That works. So, in fact, we were working on it. Francois Parsi actually published on it before us, which is cool because it's cool to see it come out. So they actually did a experiment where we knew the MADS box didn't work in the first pass, and so I was really interested in that because of we know those are heterodimers. we know that heterodimerization is kind of involved in sort of organ specification. So they ended up beating us to the punch on that. They actually did that exact Madsbox experiment. So the Madsbox, those didn't work by themselves. If you put in the correct heterodimer, it did work. The genes that you were targeting kind of made sense in terms of the different developmental organ types of the flower as well. So, but yeah, that approach does work of just sort of. This is kind of part of it too, is having that background information is important. I've been thinking through maybe approaches where you could tag the transcription factor themselves, that you might be able to do kind of a pooled approach. This is something I haven't yet got it formed enough to really give it a shot, but I think there might be ways of doing that. And I think a great example of this is like the BHLHs. They work so-so. Some of them work great, a lot of them don't work. I suspect that a lot of them are probably working as heterodimers, so if you could use that approach, then that might allow you to do it. But on the flip side, I mean, even just using protein-protein interaction maps that have already been established to then use that to then help inform which pairs you might be able to use. That's an alternative approach. It does still, it relies on kind of that pre-knowledge, but there are some of those databases available. So.
It's okay to rely on pre-knowledge.
Sure, yeah. Yeah.
You mentioned that in Arabidopsis transcription factors, you were only able to get the data for 30% of the transcription factors. It's still a lot, but I was wondering, what are some limitations of the technique? Like, cannot we just apply it in the transcription factor?
Yeah, so that was a single pass was one thing. So we did do a subset to like 100. The reviewer asked this question, so we tested 100 again, and 10% more work. So we suspect the number would have been closer to 40% if we'd done 2 passes. And so in one way, it bumps it up a little bit, but there still is this kind of remaining set that we don't have. I think it's probably a combination of a few things. I think some of them are probably folding is probably one of the big ones. It's hard to then know what happened, why it did or didn't fold correctly. I think heterodimerization actually is probably also a major one, because some of the families that we know are going to heterodimerize, like the Madsbox, like the BHLHs, those tend to have a higher failure rate than ones that may rely less on heterodimerization and more, may still, but it may also primarily rely on homodimerization. And so that's probably contributing to it as well. I think modifications and cofactors is another one. And then the last one I think is probably annotation, because these are just annotated as transcription factors. We don't really know if all of them are actually DNA binders, and some portion of them may be this. The hard part is it's difficult to unscramble which is which. I think I think the heterodimerization is one that I think we can attack, and then potentially maybe some of the modification ones through, if this other type of approach works. But yeah, that's kind of, I think, where that failure rate comes from. I would point out, I guess, in E. coli, actually, the success rate was 60%. So I think with a higher quality annotation, that's the example of higher quality annotation. Those were much better annotated. They worked much, much better at a much higher rate. So that's, anyway, so yeah.
Very interesting talk. A couple questions in the arsenic detoxification story. I think you were using the same transcription factors but different detoxification mechanisms.
Yeah.
Can you swap those mechanisms and still be functional?
I don't know. It's an interesting question. It's something we didn't do, but it should be regulated correctly, I think, and then you'd have the correct machinery in there as well. No, it's a cool idea. I think it's probably something that would have been nice to have done and thrown that in the paper, but yeah.
A broader question: you have these quite complex transcriptional regulatory networks, and I wonder how you represent them mathematically or computationally, and how you make then predictions on perturbations.
Yeah, so, I'm not heavy, it's a direction I'm very interested in going on kind of the machine learning side and the network analysis side. I do have a background in the computational side. It tends to be more on, I don't know how to just hypothesis-driven computation, essentially knocking out scripts to test specific ideas quickly and moving kind of through that direction. The other side of it, kind of more on the network analysis, I've been relying more on collaborators on that side of things. And so, I think that's, it's something I would want to build out and develop more, but at this stage, it's not my strong suit. It's not something that I have yet, the team members I need to do it, so, yeah.
Keep them coming, great questions.
Oh, that's another one.
Yeah.
Sure.
So on the DNA methylation being very common in the transcription factor regulation, have you looked at how the enzymes that facilitate the epigenetic modifications, how they are regulated? Are there interesting stories in which they are somehow regulated by the same machinery?
And I hadn't thought of that. That's an interesting direction to go. One direction I was looking at and is, specifically these transcription factors that preferentially bind to methylated DNA. In some of those cases, it looks like it requires methylation in the motif to bind, which then suggests the possibility that these regulatory pathways have to be targeting these specific sites at specific times for that transcription factor to be functional. So this is something we're interested in looking at in the multi-DAP data to see if we can see some of those relationships conserved over time, potentially even sort of tissue-specific conservation over time. And I think that would then start pointing to the fact that rather than sort of acting as these general just sort of like carpeting of methylation across a region, that there's specific targeting events, involved in TF binding and gene regulation. And so your question is quite interesting. It's something I'll have to think about, though. That's not something I've thought about or looked into, but that flip side of, how are those actually themselves being controlled would be very interesting to look at.
Thank you.
Yes.
So those 30–40% of paradoxes transcription factors that you actually could find the binding site, it's already considering the AMP or was before the AMP. So like, if you do it with the AMP for all the 1800, you would think you would pick up more binding?
No, well, it's a good point. What it is, though, is that since, the majority of the Arabidopsis genome is unmethylated, so what we see usually is it's about, the numbers right off the top of my head, I'd say sort of in terms of patch methylation, it's probably about maybe 20% of the Arabidopsis genome is methylated. So there is still enough binding sites, even without, essentially we didn't find anything that was exclusively hidden under methylated sites. And I think it may be more of a mechanism where, there's sort of globally methylation or binding sites that are unmethylated, and then at some of these transposons or other things that may be harboring, transcription factor binding sites, that the methylation is there in part to help repress that transcription factor binding and then kind of activation of expression. So we didn't find anything new in the AMP-DAP data set. What we could observe, though, was that, once again, for a small number of these transcription factors, about maybe 3% of them, they wouldn't work in the AMP-DAP. They'd only work in the methylated DAP experiment. And that indicated to us that they required the methylation to bind, otherwise they wouldn't bind. So it was the reverse that we actually ended up seeing.
So in a slightly different vein, how much have we got, or have you got, about these interactions in organelles? Chloroplast, mitochondria, human disease, powerhouses of the cells.
Mm-hmm.
What's the data, what's the horizon look like for those types of studies?
Yeah, I think this kind of information can help contribute on the side of really understanding the regulation of sort of machinery involved in some of these plastids, like the chloroplast, and
I mean the DNA binding proteins interacting with chloroplast.
It's also interesting. This is something I haven't done anything with, but it's something that Yeah, I mean, like when I was originally looking at the DAP data, I'd always have the chloroplast data as well. I'd go and look, I would see clear transcription factor binding sites in the chloroplast as well. Are they regulating it? I don't know. You know, it's an interesting question. It's something I haven't thought about in quite a few years, but it always did, you know, you kind of have this model that the chloroplast is working more in this sort of bacterial style sort of regulation, but, you know, it's also been in the presence of this, you know, eukaryote nucleus for such a long period of time. It'd be almost surprising if there wasn't some sort of.
Same is true with mitochondria, with human disease. And all that.
Yeah.
Plant development and blah, blah, blah.
Yeah. So, and I think
It's a fertile landscape.
I think so. I think, and there was definitely things showing up there. Now, it's this question: are they even there? Are they doing anything? But still, some of them looked suspiciously like they were in the right place to be controlling the gene's expression. So, but yeah, it's something I haven't dived into.
We squeezed the audience out of the question. Okay, let's let Ronan get to lunch and thank him for a very stimulating time.